Before we start today, I want to remind you that you have an online quiz that is due on Sunday by midnight, and that is going to be over chemistry. So every week you want to check and make sure, almost every week we'll have a quiz that's online. Also remember that your next in-class quiz is on Monday. So it will be only over the information that we have gone over from Monday and today. So I noticed that almost nobody got full credit for the essay question. And so you want to review that information. And specifically, you want to kind of delve a little bit deeper. So a lot of you put, you need nutrients to survive. Why do you need nutrients? And so you could say that you need nutrients in order to build tissue during growth and development, but also to maintain tissue, right? And a lot of you said that you needed water, but you didn't really have the right answer or specific answer. You say we need water because we're composed of water. But that is kind of doesn't really answer the question as why we actually need water. So we're going to be talking a little bit more about that today. Okay, so is the water molecule an example of a polar molecule or a non-polar molecule? Does it have differences in charge? Yes, right? So remember that we looked at the water molecule and that the oxygen is slightly negative because it has more protons, and then the hydrogens are slightly positive. And so this would be an example of a polar molecule because the electrons are not shared equally. And polar molecules tend to be very reactive molecules because they are going to be attracted to other molecules that have similar or actually opposite charges. Right? So a lot of the characteristics of water have to do with the fact that they are polar molecules. So if we look at the first characteristic, one is that we call it the universal solvent. So what that means is almost everything in our body that is transported in our blood, for example, is first dissolved in water. So substances are dissolved in water for transport. So our blood plasma, for example, is the part of our blood minus the cells, and that is full of substances in solution. If you look at blood plasma, if you've ever seen it, it's not red, but rather it's kind of a yellow color, and it's thick, and it's viscous, and that's because there's all kinds of stuff that is dissolved in the water in the blood plasma. So like antibodies and hormones, and albumin is a protein that is common in our blood. Clotting factors are in our blood. So in order for these substances to be dissolved in water, they have to be water soluble. And if they are water soluble, they are also polar in nature. So only polar substances get dissolved in water because the water molecules can interact with them and start to pull them apart. So when we look at proteins, for example, and enzymes that are might be in our saliva, those are examples of water-soluble substances. So when we look at a solution, we could talk about sugar solution, for example. So a solution is equal to the solvents plus the solute. So the solute is the substance that goes into solution. So we can have a sugar solution. We can have a saline solution. A sugar solution would be like where you have glucose dissolved in water. A saline solution, which you buy sometimes if you have eye issues, because if you're using contacts, for example, you put saline solution. Saline solutions are also used for intravenous applications so that it's put into your body. And that contains ions. And ions are sometimes referred to as electrolytes. And so Pedialyte is the solution of electrolytes that you sometimes give to babies that where they need, they've lost because of vomiting or diarrhea, or they're dehydrated. They have lost some of their water and some of their ions, okay? So those are just some examples of solution. So what is something that is water insoluble? Fat. Oil, right? So these are non-polar molecules. So fat and oil are called lipids. And we're going to talk about lipids in more detail today. But in order to be transported in the body, these are actually wrapped in proteins. So like if I need to transport cholesterol, which is a type of fat in my body, I would need to first wrap it in proteins, and the proteins are polar. And so they go into solution and they kind of draw the fat into solution with them. And so but we would say that non-polar molecules like lipids are water insoluble. They do not mix with water. They do not go into solution. Okay, so that's one of the important aspects of water. Another important aspect of water is that it has a high heat of vaporization. So this is where it gets turned into a gas. So it goes from a liquid to a gas. And what body function is this important in? Sweating. Sweating, right? So sweating, we have evaporative cooling. Right? So the water is able to absorb that heat energy on the surface of the skin and take with it that energy as it turns from a liquid into a gas. And so this high heat of vaporization has to do with the type of molecular bonds between the water molecules, which are called hydrogen bonds. So this has to do with hydrogen bonding between the water molecules. Remember, the hydrogen bonds are weak. Right, and so they're those dotted lines. And so they absorb energy, they break apart, and the water evaporates. Right? So that is high heat of vaporization, means it takes a lot of heat in order to turn water from a liquid into a gas. So those hydrogen bonds can absorb a lot of energy. Okay. The third thing that is important is that water molecules are cohesive and adhesive. So what this means is that they bind together, and then they also bind to other substances. So the molecules are bound together. They are cohesive because of the hydrogen bonds. And to other surfaces. So if you put a drop of water on a plate, for example, and it makes a bubble, right? That is due to the cohesive and adhesive energy that bonds those molecules together, so that the water doesn't form flat, it doesn't go flat, it actually forms a bubble. And where this is really important is in our capillaries. So the capillaries are really small blood vessels, right? And we have what is called capillary action. And you can use straws, for example, to demonstrate capillary action, so that when you put a straw in water and then you put your finger over it and you draw it out, there's water being held in the straw, right? So the water molecules are kind of clinging to each other, but they're also clinging to the side of the straw. And this is the same thing with capillaries. So sometimes in medicine, we use capillary tubes. You can prick your finger and then use a capillary tube to draw the blood up. into a really, really thin tube. But the capillary action just keeps the blood moving through the capillary so that it doesn't stop when it gets to lots of resistance because the vessel is so small. Okay. So that is the idea of capillary action. And then the fourth thing is it is important in chemical reactions. So it participates, water participates in chemical reactions. Okay, so we talked a little bit about the different types of chemical reactions that we have. And so remember we talked about decomposition. So this is a form of catabolism or breaking apart of molecules, but this is also called hydrolysis. So if you think about what this word means, when you start to learn these roots, it becomes easier to understand them, so you don't have to memorize the individual words. As long as you know what the roots mean, you know what the word means. So hydro means water. Lysis means to break apart. And so water molecules help to break apart proteins, they help to break apart sugars, and they actually bind to the molecules after the molecule has been broken apart. And so that is called decomposition. Now, synthesis is the building of molecules, and it has another term, which is called dehydration. So dehydration is the removal of water. Right? So water, rather than water being used to break apart and adding to the molecules, water is removed from the molecules during synthesis, and that is called dehydration. So I'll show you some pictures of those as well as some of these other examples. So let's just look at universal solvent first, okay? So this is my water molecule, and this is salt. So remember that salt has positively charged and negatively charged ions in it. So the sodium chloride, if you put salt in water, it will be pulled apart by the water molecules interacting with it, and it will diffuse. So diffusion is just the movement of a substance from a high concentration, like a grain of salt, and it will move, and there'll be molecular movement until the salt is equally distributed through the water, and then an equilibrium is actually produced. Okay, so that's salt going into solution. This is an example of the chemical reactions. So if you have two monomers, and we'll talk about what those are in a little bit, but if you have two things that you're going to put together, you're going to remove the water. And so notice that there's OH and OH on this side, and we can take the water out of that, and we form a new chemical bond, a covalent bond between those two molecules. So energy is stored and water is released. We also can use water to break stuff apart. And so this would be hydrolysis, where we add water, we break up covalent bonds, and then we get water, the atoms being attached to the substances that are subsequently produced. So this is all the reasons, kind of significant reasons why we have water in our body and why we think that water is essential for life. So when we're looking at other planets, we say, oh, if there's not water, then there can't be living things. So it's kind of interesting, this idea that water is so significant to life. Okay, so we are now going to start on the next chapter, and this is organic chemistry. So what we have been talking about so far is inorganic, so versus inorganic. So water is an inorganic molecule, and we kind of define this by the fact that it does not contain a carbon skeleton. Salt is inorganic, hydrochloric acid is inorganic, sodium hydroxide would be inorganic. Okay. Oxygen gas would be inorganic. So organic chemistry means that the molecules have a carbon skeleton. And this skeleton can be a chain. So it could be a long chain of carbon atoms. And you can have other atoms covalently bonded to them. So you could have like hydrogen and oxygen and nitrogen and sulfur all bonded to the carbon in different orientations. But you can also have not just chains, but you can have rings. And so sometimes carbons form rings. And so if I drew my ring right here, okay, so at every juncture in this line, there is a carbon atom. So I'm just going to write, there's a carbon at each junction. Oftentimes when we're drawing organic molecules, we leave the elements out of it. So we would draw that, but we wouldn't put carbon in there, right? We would just assume that you knew that that's an organic molecule. Okay. So then we would have other elements covalently bonded to that. So if we look at some examples of these, and this is pictures in your book, okay? This is a long chain of carbon. This would be like, for example, a lipid. And one of the reasons why I know it is lipid because it's non-polar, because there's an equal distribution of charge here. This is like an amine group. So proteins have lots of nitrogen in them. And so that is like an amino acid. And this is like a sugar. And so notice I drew that this was what I drew before. And the shorthand would just be eliminating the carbons. And you can kind of assume if carbon is present, then you would draw it that way, okay? And so here's another diagram that shows the different amino acids. And so here's my ring of carbons shown there. So just don't be confused if you see weird shaped structures and you're like, well, I don't see any carbon. The carbon is just not drawn in there, but it is there. Okay, so the first group of organic molecules that I want to talk about are the carbohydrates. So carbohydrates are essentially sugars. And so they are really important in energy. Right? So we obtain food not only for nutrients, but also sometimes for energy. And carbohydrates generally are a fast way of obtaining energy. The problem with them, what we'll talk about maybe, is that they oftentimes, regulating sugar levels in your body is oftentimes difficult. So if you take in too much sugar all at once, if your body cannot regulate it, then you get diabetes. And that is just a really bad kind of cascade of metabolic events that can lead to really bad things, okay? So you have to be careful when you take in energy. So sometimes we can break down carbohydrates into what is called the monomer. So the monomer is the basic unit of how carbohydrates are built. And in carbohydrates, the basic unit is called a monosaccharide. Mono means one, but what does saccharide mean? Is that a sugar? Yeah, but what is it like if I said she had a very saccharine personality? Sweet. Sweet, right? So that means sweet. So that's a single sugar, okay? So that's the monosaccharide. So some examples of monosaccharides that you are familiar with are glucose, fructose, which is like fruit sugar, galactose is another one. Okay. So those are the monosaccharides. When we look at the disaccharides, these are the ones that have two monosaccharides. So they're disaccharides means that they have two monomers. So di means two. And so an example would be lactose. And where do you get lactose? Milk, right? So you have milk, right? And this is a disaccharide, so that if I break this down, I can produce glucose plus galactose. Okay, so would that be a synthesis or a decomposition? Decomposition. Would it be hydrolysis or dehydration? Hydrolysis, right? So we have to have water, so I can put water in there. And then we also have to have an enzyme. And so in this case, it is called lactase. Okay. So remember that ACE ending means that that is the enzyme that breaks down lactose. And that is in our small intestine. So that enzyme is produced by the cells in our small intestine. And it's kind of interesting, an example, because lactose is found in milk, and the vast majority of adult mammals do not drink milk, right? So when we look at milk, this is nutrition, has lots of different things, for newborn mammals, or young mammals, right? So if a newborn mammal is going to be able to digest lactose, they need to produce lactase, right? If we, upon weaning, right, we stop drinking milk. Stop drinking milk. Okay. So then we stop producing, in the most cases, we stop producing lactase. However, it's kind of interesting because when we look at human populations, there's one major group that seems to have the ability to digest lactose even as adults. And so in humans, the ability to digest lactose is dependent upon the population in which you are from. Okay, so specifically, in relationship to the United States, it's the Northern Europeans. And they evolved over time to keep producing lactase. Even as adults. Okay. This ability to produce lactase as an adult has a name, and it's called lactase persistence. Okay, so you probably don't hear much about lactase persistence because actually in the United States, it seems that most people have some ability to digest milk as adults. And so you hear of it as being what? It's not lactase persistence. If you can't digest lactose, what's it called? Lactose intolerance. So in adults, in adult mammals, this is the norm. So if you have the ability to digest lactose, you are rare. You are a rare mammal on this planet. And it is believed that the Northern Europeans have that ability because they live up in colder climates. And over the winter, they relied upon dairy animals to produce milk so that they could survive in these cold, harsher climates than, say, for example, if they lived in Asia or if they lived in Africa. And so there's actually another group of humans that can digest lactose, and they are a group of herders in Africa. So there's like two groups of tribal groups in Africa where one group is a herder and one isn't. And the herder, the one that keeps the milk-producing animals, are lactose or lactase persistent. So it's not just in the Northern Europeans, there's actually a tribe in Africa as well. So the idea here is that lactose intolerance is an adaptation so that you can eat milk. But in some other cultures, they have made milk more digestible. And how do you make a product more digestible to a person? Pasteurization. Nope, pasteurization actually kills the microbes. So that actually protects us from bad microbes. But how do we treat our milk products so that you can digest them a little bit easier? Nope. Enzymes, what produces the enzymes? What's cheese and yogurt? Dairy products that have been fermented, right? So like in Asia or in India, they might actually probably in India, excuse me, in India they use yogurt, right? And so they can't digest lactose, but they can get nutrients from the yogurt. And the yogurt is where you let the yeast digest the milk sugars, and then they produce substances that we can't absorb. So we can fermented milk products increase their ability to be digested. Yes, so if it's rotten, we can eat it.
So you have really good hearing.
Yeah. Cheese, right? Blue cheese.
Ew.
Blue cheese, yum. Okay. Swiss cheese.
That's good. Yeah.
Okay, so cheese and yogurt. In Russia, they have a drink that they serve to every child at school, and it is called kefir. And it is very nutritious, and it's kind of interesting. You can make your own kefir at home, and what you do is you get milk and you add some bacteria and yeast to it, and then you let it sit on your counter for like a week, and then it becomes really frothy, right? It becomes really frothy. So it's almost like it's really cool because it's almost like carbonated milk. Okay? And you can buy kefir in the grocery stores now. It became really popular. Yeah. So sometimes you can also remove the lactose. So if you're lactose intolerant, you can have lactose free. And then occasionally there are babies that are born that cannot digest lactose. And so then you have to give them lactose-free soy products, right? So you have to give them maybe formula that does not have lactose in it. Now I want to point out that this is a little bit different than an allergy. So this does not cause inflammation. It does not cause an allergic response. The reason why it feels so bad if you're lactose intolerant is that you have microorganisms that digest the lactose and then they produce gas. And that gas causes intestinal cramping if you drink too much lactose. Okay, are there any questions about that idea of lactose and lactase and breaking it down and using it for nutrients?
It's better to eat fermented stuff than regular.
Yes, yes.
Yeah.
I actually just made, for the first time, kimchi on my countertop. That was kind of cool. So you just take vegetables, you chop it up. Well, it's just like the kimchi in the grocery store. You chop it up and then you add some bacteria to it. Or you can just do wild fermentation because there's bacteria in the air. But then you just let it sit on your counter for a week and it ferments and it just tastes like sauerkraut. Yep. Okay. Okay, so this is the diagram of the monosaccharides. So we talked about glucose, fructose, galactose. We'll talk about these more. These are actually in our DNA and in our genetic material. So DNA has sugar in it, and it's called deoxyribose, and our RNA has ribose in it. So those are also monosaccharides. So I wouldn't make you obviously tell the difference between these, right? But if you see just a single ring like that, you would know it is a monosaccharide. So here's an example of a disaccharide. So this would be sucrose. So sucrose is another one. So sucrose is table sugar. And so it is fructose plus glucose. So you have probably heard that high fructose corn syrup can be really bad for you. And they're actually trying to ban it because of obesity and diabetes epidemic. But if you look at some of the new sodas, they have what are called cane sugar. And so cane sugar would be sucrose. So this is better for you because it is a disaccharide. So it takes a little bit of time to break it down, and your body can metabolize it and break it down. And so it doesn't cause such a spike in your sugar levels in your blood. High fructose corn syrup, actually, because it's fructose, cannot be metabolized by all the cells in your body. It can actually only be metabolized by your liver. And so drinking soda pop is really bad for your liver because it has to metabolize the fructose. So it puts a little stress on the liver. And then it also would cause a bigger spike in sugars, in your blood sugar levels as well. Okay. Okay, so if you have many of these monosaccharides bound together, then you have a polysaccharide. So this would be many monomers, many monosaccharides. So we can look at two different types of polysaccharides. We can look at those that store energy, so storage of energy. So starch, for example, is the way that plants store energy. So if you think about potatoes, potatoes are the roots. The plant dies during the winter, the potatoes, if they don't freeze, can overwinter, and then they can sprout from the energy. Sometimes potatoes sprout in your cupboard or on your refrigerator, right? They're trying to grow and reproduce. Okay? We do not use starch. We use glycogen. So this is in animals, and we do not obviously have roots, but we store this in our liver and in muscles. This is in muscles, so stored in the liver and the muscles. And that's because the muscles specifically need to use this energy to break it down to produce energy to move. Okay, so that's storage of energy. We also have in some cases, structural polysaccharides. We do not use protein necessarily for our bones, but we do use it, actually we do use it in our bones, never mind. We use it animals, let's talk about plants first. They have cellulose. So these are plants. So the cellulose is the fiber. So fiber is indigestible to us. So if you eat celery, for example, it's got so much fiber in it that it actually takes more energy to digest it, to actually break down everything and pass it through your body than you actually get from eating it. Okay? So some organisms, however, can digest cellulose by using bacteria in their gut. So like horses and cows can digest grass and use it as nutrients because they have bacteria that digest the grass, and then subsequently they digest the bacteria. So they actually ferment the cellulose. So we cannot break down fiber, but fiber is really important because it keeps the digestive tract and stuff moving through, right? So they've discovered that if you don't have a regular bowel movement, if you are constipated a lot, then you are actually at a higher risk for developing cancer, specifically like colon cancer, because toxins stay in your digestive tract too long and they can actually damage the cells. And so being regular has to do with like having fiber in your diet and being able to get rid of the waste material. Okay, so in animals, we have keratin and not cellulose, keratin and, just collagen. There we go, collagen. And these are words you oftentimes see on skin creams, right? And oftentimes you see them in your shampoo, right? This contains protein, keratin, right? And so when we look at keratin, keratin is the protein that is found in your skin, hair, and nails. So your hair is actually dead cells that are filled with keratin. Your nails are also dead. The outer part of them are dead. They have actually a living cuticle. So the moon on the beginning of your cuticle is actually live cells that are undergoing cell division and forcing the nail to move and grow longer. Okay, so collagen is found in the dermis of the skin. So that's underneath the skin, the outer layer, the epidermis. And it is also found in your bones and in your cartilage. So the reason why bones and cartilage are white is because of the collagen fibers they have in them. So those are structural polysaccharides. Wait a minute, I'm confused. Oh, I knew that wasn't right. Okay. I'm talking about proteins and I'm talking about polysaccharides as if they're the same thing. Okay, these are, just right there, sorry, these are structural proteins. Okay, not polysaccharides. Oops, sorry. I knew we didn't have polysaccharides in our skeleton. Okay, so cellulose is a polysaccharide. We actually, like insect exoskeletons are actually made of sugar as well. We cannot digest them. So those are structural proteins. Okay, that was embarrassing. Okay, next group, lipids. So are lipids polar or nonpolar? Nonpolar. They are also water insoluble, and that has another name, means that they are hydrophobic. No, we're on the next one. So this is the second one. So the first one was carbohydrates. The second major group are the lipids. So hydrophobic means that they are water fearing, right? So they are water insoluble. Okay, so lipids are another group of organic molecules.
So I'm gonna ask. So everything we were talking about here.
Just with the animals?
Was the carbohydrate section?
Yes, so everything that was with the animals, under keratin and collagen, those are proteins, not polysaccharides. So I skipped ahead in my lecture.
Cellulose.
Cellulose is a polysaccharide.
That is a polysaccharide.
That is a polysaccharide.
That is the only one that was.
That was the only one that's the polysaccharide. Yeah. So everything else was carbohydrates. So monosaccharides, disaccharides, polysaccharides. It's just I talked about proteins right in the middle there. Okay, so these are hydrophobic and they include fats, which are animal, and oil, which would be in plants. Okay, so this is kind of a generalization. The plants like corn oil, peanut oil, sunflower oil, right? They are the oils. We'll talk about what the difference is in a minute. Animals have fats. And so the difference between these has to do with whether or not they are saturated or unsaturated. So in animals, the fats are saturated. Okay, and we'll talk about what that means in a minute. And they are also, they're saturated, and they also tend to be a solid at room temperature. So if you think about lard, Crisco, that is a saturated fat. Now, sometimes the house gets too hot, right, and it turns liquid, but generally tends to be saturated. Okay, plants.
Where would, so, I mean, soy, like, Crisco, for example, is mostly it's vegetable oil, right?
Yeah, so they've actually hydrogenated it.
Okay.
Yeah, so I'll talk about that in a minute. So then plants are unsaturated. I guess I was wrong. There's a difference between lard, which would be animal fat, right, and Crisco.
Right.
So when you like save your bacon grease, that would be fat. That's lard, yes.
So that would be in the saturated category.
Yes. So unsaturated would tend to be a liquid at room temperature. Now, one example that's not is, which has become really popular, is coconut oil. So coconut oil tends to be more of a solid, right? And they argue that it's good for the body because it can be used and broken down, and it's used to produce energy really efficiently by the brain. Okay, so that's kind of its a generalization. Okay, so let's look at the saturation. So saturation refers to the number of hydrogen ions that are covalently bonded to the carbon skeleton. So I'll just put my hydrogen ions there. Covalently bonded to the carbon skeleton. So if it is saturated, there's the maximum amount of carbon. So if I were to draw 3 carbon atoms, and I put hydrogen here, here, and here, that means that it is saturated, okay? So that means that it has kind of the maximum number. So here it would be more carbon bonded behind and in front. Okay, so unsaturated would mean that we have double bonds. So for example, we could put a double bond in there, right? And now this one, if it likes to have 4 bonds, it would have hydrogen here. This one would have hydrogen here and here, here and here, okay? So this means that it is unsaturated because it has a double bond instead of a hydrogen atom covalently bonded to it. Now, if we look at their structure, we can kind of see how or why we have the difference between a liquid and a solid. So a solid is where the molecules are able to tightly pack. And here you can see that they would be able to, well, they're able to more tightly pack than if they are unsaturated, which makes it unsaturated more likely to be a liquid. Okay. Oops. Okay. So notice how the saturated is like a straight line. So if you have a bunch of these together, they pack really tightly. The unsaturated has a little kink in it where there's that double bond, and so they don't pack tightly, so they are more likely to be a liquid. And that's kind of the difference. You don't need to know that, but that's the reason for the difference in the states of the liquid versus solid. Okay.
So are unsaturated easier to digest?
They are better for us. I don't know if they're easier to digest. They're better for us because they are less likely to clog arteries, right? And they also affect our cholesterol. So cholesterol is a different type of lipid, which we're going to talk about in a minute. Yeah. So I don't know if they're easier to digest than the saturated, but they're better for our cardiovascular health. Okay. So let's look at the hydrogenation of corn oil, which is Crisco. Right? So look at that word, hydrogen. This means that you're breaking the double bonds and you're producing and you're adding hydrogen to the structure. So that's why it's called hydrogenation of corn oil. And so we say that it's hydrogenated fat. Okay. Now, hydrogenated fat also has a bad effect because it is an example of a trans fat. Okay. And trans fats would be like Crisco, but it would also be like margarine. And anytime you use hydrogenated fat or corn oil to, even if you're using it to fry stuff, it could be trans fat. So you remember there was a time where people, if you were getting fast food, you know, they might say, we do not use trans fat or whatever, right? So the trans fat is a form of hydrogenated plant oils. And it has a problem in that it increases cholesterol levels. We'll talk about cholesterol in a minute. So it is more unhealthy than simply consuming saturated fats. So for example, I would say if you had to choose between margarine and butter, butter is more healthy than margarine because margarine is generally a trans fat. So even though the butter is saturated, it is not a trans fat. So it's going to actually not affect cholesterol levels as much as trans fats are. So that's why they were having this big controversy over whether or not we should ban trans fats in our diet just to keep people healthier so that we don't have to pay for cardiovascular diseases that lead to stroke and heart attacks. Okay, so in your book, actually, this, I'm not sure if this is in your book. This might be from somewhere else. But unsaturated fats, these are called the good fats. Saturated fats are mainly found in animal products. And then the trans fats, they're trans, and they've hydrogenated because they increase their shelf life, but they are bad because they elevate cholesterol levels. So fats and oils have a name, and together they are called triglycerides. Oops. So the triglyceride is the term for both fats and oils. And the reason why they're called this is tri means that there are 3 groups of long chain fatty acids. So that's be the chain of carbon coming off, right? And then we have a glycerol molecule located here. So you need to be able to, like if this was, if I showed you this picture, right? You would say, I know that is a triglyceride because there's 3 chains of carbon, tri, and then there's this one structure right here, which is a molecule called glycerol. Okay. So triglycerides can be used to store energy. Oops, to store energy by us. Right? It can also be used for insulation. And specifically, we can talk about subcutaneous fat. Right? So sub means underneath our skin. So when we get, love handles, for example, that is subcutaneous fat, and that actually helps with insulation. Mammals that are aquatic, like whales and dolphins, have lots of insulation, right? Because they're trying to protect themselves from losing heat to the water. Okay. We can also talk about how it cushions organs. So specifically like the kidney, which is located near the back of the body, is cushioned by a layer of fat. And then there's also fat around the heart. And then sometimes we can get what is called visceral fat, right? So this is wood cushion organs, but it would be visceral fat. So viscera is organs, right? But this is in the body cavity. So this is the beer belly, right? So, you know, I had a stepfather who had a huge belly, right? And he would say, come on, punch it, it's all muscle, right? And the reason why it was firm, right, it wasn't flabby, it was all muscle because the fat is underneath the muscle, right? So you have the muscle, and then you're putting fat into your abdominal cavity, and it's making your belly look like that, right? They say there's no fat, it's all muscle, because they can't feel the fat because the fat is underneath the muscle. So they say that visceral fat specifically is worse for cardiovascular health than subcutaneous fat. And so if you have that characteristic of that big belly and it's hard, that means you're at a greater risk for heart disease than if you have lots of subcutaneous fat. And men tend to have more visceral fat than women. Women tend to store their fat subcutaneously. So those are triglycerides. Okay, so another group of fats, if we look at other lipids, one of these is called the steroids. So does anybody know what hormones do we have in our body that are considered steroids? Testosterone. So what do females have? Estrogen. And these are actually all derived from cholesterol. So cholesterol is also a steroid. Cholesterol is also necessary for the production of vitamin D, but it is a precursor to testosterone and estrogen. So these have a characteristic shape where they have 4 carbon rings. So their shape is much smaller than the triglycerides because they're not storing energy, right? And it is very characteristic, the 4 carbon rings. And testosterone and estrogen look almost identical. And actually, our bodies can convert testosterone to estrogen and vice versa, right? So in my body, testosterone is produced by my adrenal glands, and estrogen is primarily produced by my ovaries. So both males and females have testosterone and estrogen. It's just the relative amounts that make a difference. Okay. So if we look at a diagram that shows this, right? This is my 4 carbon ring right here. Okay. So actually not that one, this one. So this one right here shows the 4 carbon rings, right? And remember that there is carbon here, it's just not drawn in, right? And that's the characteristic shape. Most hormones are not steroids. So we know a lot about steroids because they're important in sexual development. And they also, we'll talk about this, but you can actually also get steroids through the skin. So you could use estrogen cream or testosterone cream, and it goes right through the surface of your skin and into your body. Whereas, like if you put another hormone on your skin, it wouldn't be able to be absorbed. Okay. So the next group of lipids are really important because they make up our cell membranes, and these are called phospholipids. Okay. So we have triglycerides, we have steroids, and then we have phospholipids. Phospholipids have a characteristic shape where they have a head end, and then they have 2 tails. So this is what is referred to as the head of the molecule, and this is the tail. Now this is actually not this is not the molecular shape, but this would be like the 3-dimensional structure that it forms. So this is not where the carbons, you don't see carbons in a circle here. Okay. So this molecule is said to be amphipathic. which means it has a two natures. It has two natures. And so the head is hydrophilic. So that means water loving. So do you think the head has a charge to it or not? Is it polar or not? It is, right? So it has a little charge to it. So I'll put a little positive there. And the tail is hydrophobic. So it is nonpolar. Well, the cool thing is if you put a bunch of these molecules into a beaker of water, they will arrange themselves in a lipid bilayer. And so it actually looks something like this. So here's my layer of phospholipids. Right? And they actually create these spheres, right? So this would be like if you just threw a bunch of these in the water, the tails are repelled by water, so they tend to be attracted to one another because they're nonpolar. The heads are attracted to the water, so there would be water on the inside and there would be water on the outside. Okay. So they arrange themselves in this way without any energy input. And this is actually what makes up the plasma membrane of animal cells. Okay, so this would be the plasma membrane of animal cells.
Is this the same as the sperm concept?
No. The sperm and the tail is different. The tail is, it's not the same. So the tail of a sperm is protein. So if we look at the tail of this molecule, okay, this is a phospholipid molecule right here. Okay, so the head has a positive nitrogen. I don't know if you can see that, but there's a positive nitrogen right there. But notice that it is just made up of atoms, right? And the tail, when they put these two dotted lines right there, that means it's actually much longer carbohydrate chain. So there's just a long chain of carbon molecules. And so it's not the same thing as the sperm, because we'll talk about the sperm, but the sperm has proteins that form the flagella. And so it's different. And it's not, they're not hydrophilic or hydrophobic. Okay. So we also have in our body wax. So as another example of a lipid, and can anybody tell me where we produce wax? Ear. Yeah, I love earwax. This is so interesting. Okay, it is cerumen, and it is yellow, and it's smelly, I think. And I did a little research on earwax because I have lots of earwax, and when I was a kid, they would always have to flush it out. And what's really interesting is that wax is hydrophobic, right? So it's not soluble in water, and what it does is it actually migrates. So the cells produce the wax, and then the cells migrate, and they carry the wax to the outside of your ear canal. So theoretically, the cells should be able to move the wax out, right? But sometimes if you use those Q-tips, which they tell you never to use, right? Don't use your Q-tip. If you stick it in there, it actually pushes the wax too far in, and then it cannot move out, right? And the thing that I found most interesting about this earwax is that when you look at aquatic mammals like whales, they do not get rid of their earwax. And so as they produce it, it just accumulates in their ear. And so they can find these little cores of earwax in a whale that are like 24 centimeters long of earwax. And then they can look to see if there's any toxins in the earwax. And so toxins are oftentimes soluble in fats. So like DDT and PCBs, and sometimes they can see exactly when in the earwax toxins were present because they got put into the earwax. And so it's almost like tree rings. So the cerumen has antibiotic properties, right? So it has antimicrobial, not antibiotic, but antimicrobial properties. And there's actually a genetic component to it because like Europeans tend to have wet earwax and the Asian populations tend to have dry earwax. So there's seems to be some variation in the genetics behind earwax. Okay, so those are the lipids. So now we're going to move to proteins. Okay, so this is where I got confused. Okay, so proteins are composed of amino acids. We have 20 different amino acids in our body. And they can be put together in any order. So the amino acids are put together in a specific order, and you get a chain of amino acids. And so oftentimes what they do when they're showing protein structure is they'll draw the amino acid as a circle. So it doesn't really look like a circle, but so this would be like the 3D structure again. Circle, 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 etc., right? So when you look at the bonds between the amino acids, these have a specific name, and they are called peptide bonds. So peptide bonds occur between amino acids. And this explains why sometimes proteins are called polypeptides. Okay, so this is the same thing as a protein, a polypeptide, same thing as a protein. So when we start talking about how proteins are synthesized by your body, that will be important. Okay, so when we look at these produce very biologically active molecules. So they do lots of things, okay? So for example, we have enzymes. And enzymes, I always have to do this because people get confused. Enzymes are not alive. And the reason why people get confused is that sometimes you see food and it says living enzymes, right? They're not alive, they're active or not active. Okay, so these are not living. They're active or not non-active. Okay. So sometimes it's actually good to eat food that has enzymes in it. So like if you eat raw vegetables and raw fruit, the enzymes are present, right? If you pasteurize milk, then you remove all the enzymes. But breast milk would have active enzymes in it, and breast milk would allow the newborn to more thoroughly digest the milk, right? So enzymes are examples of biologically active molecules, okay? Most hormones are proteins. So even, and this is kind of confusing, even growth hormone. So this is the hormone that causes bones to become longer in children. So it causes their bones to grow and them to become taller. Okay, so that is actually a protein. That is not a steroid. That is called that would be a protein hormone. Okay, antibodies, which have an immune function, are also proteins. Okay, and we could also talk about clotting factors in the blood. So this allows blood to clot, right? And they are proteins. Okay. So all of these proteins that I've just labeled or I've just described are examples of globular proteins. And globular proteins have a complex 3D shape. So think like a glob. So what that means is that that chain of amino acids has to fold. And so that folding has to do with hydrogen bonding between different parts of the molecule, which causes the polypeptide to fold, right? So we have folding of the polypeptide. All of these that I just talked about are soluble in water. Right? So they can go into solution. The clotting factors actually come out of solution in response to an enzyme, okay? But in most cases, they are in solution, okay? And I'm going to say that they are relatively unstable. And what this means is that temperature and pH changes can lead to denaturation. Okay. So if I drew my complex three-dimensional, this is my globular protein, right? If I denature it, it just turns into a long strand. Okay, so this would be denaturation. So sometimes heating can do that. So for example, when you look at an egg, right? First it's liquid, you're like, it's really thick, viscous liquid, right? But then you put it in the pan and you add heat. The reason why it turns into a solid is that the proteins come out of solution and they denature, right? And so then they're not soluble in water anymore. And you can't take and go the other direction, right? So you can't take a solid egg and turn it back into a liquid. Actually, I think they might have just done that years ago. But anyway, in general, it doesn't happen, right? Okay. So that is the process of denaturation. And interestingly, you can actually cook an egg by putting it in acid, right? So you can cook the egg. It will denature in low pHs as well as high temperatures. So if we look at this three-dimensional structure, okay, this is the peptide bond between two amino acids. Notice that the amino acids have nitrogen in them. That'll be important when we start talking about waste products, because when you break down proteins, you have to produce more urea. So you actually have to urinate and get rid of the urea because of the proteins that are found, or the nitrogen that is found in the protein. Oops. Okay, so this is my strand, my polypeptide strand. And so you can see here that different parts of it form little, they get attracted to other parts. And so this would be an example of a hydrogen bond, for example. This is another type of bond, which is called a sulfur bond. You don't need to know that. And some ionic bonds are located right there. So in your book, they have different degrees of folding. So this shows the hemoglobin molecule. The globin part, hemoglobin is what carries oxygen in your blood. And so the globin part is protein and it is highly folded. Okay, this would be the iron part of the hemoglobin. And that three-dimensional structure is what determines its activity. Okay, so we'll stop there for today, and we'll talk about a little bit more about proteins, and then we'll talk about nucleic acids. Don't forget about your online quiz this Sunday.